Welcome students. This is a continuation class for the 5th semester B Pharmacy students. Last class we have discussed about the essay questions and from December 2023, and I would like to continue with the short questions for 5th semester in December 2023 KUHS University question paper. It will be interesting for you to write down and as well as listen and to know how to write the exam, the answers which you have already come across. It will be interesting for the students to listen to this type of lectures and to know how to write my university examination. So you see, last class when we were discussing about the Senna, they have asked in the essay question about senocytes, how will you isolate and estimation. They used to ask 5 marks separately also, but in essay question we have discussed about glycoside definitions and classifications and androgen glycosides. But I told you to specifically to write about the extraction and isolation of senocytes from senna. But today, before going to the short answers question, I would like to tell what all the things have to be added in that essay question. Here, like senocytes, in the second essay question in December 2023, they have asked about senocytes isolation from senna. So in which students have to write about senocyte structures, and senocytes are obtained from the dried leaves of Cassia senna, Cassia angustifolia, family Leguminosae, and the structure you should write clearly the senocyte A and senocyte B and senocyte C. How from the senna leaf, and they have the glycoside linkage, O glycoside linkage of D-glucose in the two structures. Clearly it is drawn and shown to you, it should be mentioned. Senocytes are dimeric androquinone glycosides, meaning they are derived from androquinone, quinone, double bond O, quinone, N-O-N-E. You can see the double bond O, quinones, and have two sugar molecules attached as shown in the picture and the diagram which I am highlighting. You can see the D-glucose molecule in senocytes, how they are made into, and also they have the geometrical isomers of A and C and B and D. You should know about the senocyte A, B and C, what is their relation of geometrical isomers. They are primarily found in the leaf and pods of Cassia angustifolia and Cassia acutifolia plants. Senocytes, especially senocyte A and senocyte B, are used as stimulant, laxative. They can stimulate the movement of the bowel. They work by irritating the colon, increasing peristalsis movement and reducing water absorption. Industrial production, prepare and initiate extraction. Senocytes or senoleaves are dried and grounded or comminuted into powder. The powder is extracted with solvents like benzene, methanol, or a mixture of methylone and toluene. The extract is filtered and the residue, that mark, is separated. Further, the second step, the extraction and the concentration. The mark is further extracted with 70% methanol and other suitable solvents. The extraction are combined and concentrated often under vacuum. You should remember about the vacuum filtration which I shown in your lab about Buckner funnel, in industrial large production, they will be using vacuum filtration to remove the solvent. Purified and isolation is done. Precipitation with calcium chloride can be done. A solution of calcium chloride in methanol is used to precipitate cyanocytes from the extract. Chelating calcium cyanide complexes. pH adjustment and separation. The pH of the extract is adjusted with ammonia, and the precipitated cyanocytes are filtered and washed with methanol. Now recrystallization. Cyanocytes A and cyanocytes B are further purified through recrystallization with different solvents like glycomonoethyl ether to enhance purity. Isolation of cyanocyte A and B. Cyanocyte A is obtained by treating the calcium cyanocyte precipitate with acidic gluconic acid, while cyanocyte B is isolated by using methanolic hydrobromic acid. So the chemicals you should remember. Drying and storage. The isolated cyanocytes are dried and stored in a desiccator for long-term preservation, like this desiccator, to prevent the atmospheric moisture should not be absorbed by the product. Estimation. Usually industrial production and estimation of the component according to the unit 5 is very important. Cyanocytes, the active ingredient of senna, are typically estimated using analytical techniques like HPLC, high performance liquid chromatography, or thin layer chromatography, TLC, after extraction from senna leaves or pods. We can see this is a picture to show you. For TLC plates, separation of the compound, identification of the compound with original cyanocyte A and B can be done by TLC, HPTLC, HPLC. HPLC is favored for its precision and ability to separate and identify by peaks in a very quantitative estimation can also be done, like A and B, cyanocyte A and B individually. While less precise than HPLC, TLC can be used for qualitative analysis, and the preliminary step is very detailed quantification can be done also by HPLC. Even HPTLC can also be performed. Pharmacognostical application of cyanocytes. We know that cyanocytes primarily found in senna plants are utilized in pharmacognosy as a potent stimulant laxative for the treatment of constipation and preparation of bowel before surgery or medical procedures. You can see the picture how the laxative and cathartic. Laxative will be fully formed fecal matter from the rectum, but evacuation of entire column as a liquid form is cathartic. The dose is increased, then you can use as a cathartic also. They work by irritating the colon, large intestine, increasing peristaltic movement and reducing water absorption, ultimately leading to the bowel movement. Now we are going to discuss about the short notes, 7 short notes which we are coming across in the December 2023. We are not going for detail about all the 7 questions in today's lecture. We will continue in the next lecture also. So it will be interesting for the student, will be knowing about part, listening, at least in 1 hour, please spend to listen your revision, your classes. They have asked in 5 mark question, 7 questions. Write a note on lignans and corticoids or or you can say carotenoids, not corticoids, carotenoids. The lignans is in the chapter, it comes in phenolic and another chapter, and carotenoids will come in another chapter in the units. So we will discuss about that. But anyhow, you should write specifically 5 mark regarding what is ligand or ligands or lignans, you can say, not ligands, lignans, or what is its application for the pharmacognostical application, what is its pharmacological uses, therapeutic application, and carotenoids, what is its therapeutic application, what is its source in plants and animals. And number 4, explain tracer technique. In the first unit which we have discussed about tracer technique, 5 mark question. But they used to sometimes essay question. So little elaborately I have explained in this answer. So today's lecture can be detailedly 5 mark, only 2 we can explain, because I have explained little more if they ask 10 mark. But here only short manner we can write for 5 mark question. And the next 5 mark question, describe the anatomy of digitalis, describe method of production and essential, or not essential, estimation of Taxol. It's a very good, interesting Taxol anti-cancer drug. So how will you estimate and how will you produce from the plant source? They're asking for 5 mark question. And differentiate between Sumatra benzoin and Sima benzoin, the 2 species which are available as a resin and a very interesting medicinal value. What is their difference by means of their botanical source and chemical constituent and uses? All these things can be explained. Differentiate, 5 mark question. And number 8, describe the method of isolation and analysis of podophytotoxin. So the isolation and extraction in this question paper they have concentrated more. Isolation and analysis of podophytotoxin. And number 9, the question they have asked, explain the industrial production and estimation of vincristine and vinblastine. They have concentrated in this chapter more how you can isolate, how you can estimate this vincristine and vinblastine. The students should know about the chemical structure and the chemical nature of the compound and presence in which source, and how to analyze them, how to estimate them, details should be given. First we will discuss about, write a note on lignans and carotenoids. You know, lignans comes here in our chapter number 4 or 5 unit, phenyl propanoids and flavonoids derivative. First one, lignans, T, ruta. But today we will discuss about lignans. And in iridoids and other terpenoids, another chapter by naphthoquinones, they have given gentian, artemisia, taxus and carotenoids, in which they have asked the question 5 mark carotenoids. So we have to explain about lignans and carotenoids. Now we will see about what should be written for lignans. Lignans, the term lignan was first introduced by Haworth, 1948, to describe a group of dimeric phenyl propanoids. Phenyl, phenyl, 6-membered ring, phenyl, benzene, that is phenyl with OH, and propanoids, phenyls, propanoids. In the name itself, you should remember the imaginary chemical structure should come into the pharmacy students, which were 2 phenyl propanoid molecules. Phenyl propanoid molecules. A propanoid with a phenyl group, a benzene with OH group. That's what your imaginary diagram should be there. Molecules are attached in the central carbon atom with carbon 8. 8 carbon atom. So you can see lignans are here, just I have to pull it down, otherwise you cannot see completely. Here you can see here lignans are a subgroup of non-flavonoid polyphenols. Polyphenols, OH group in polyphenols. They are widely distributed in plant kingdom, being present in more than 55 plant families. That's why we have taken as a consideration for lignans, where they act as an antioxidant and a defensive molecule against pathogenic fungi and bacteria. So they are very important therapeutic activity against very pathogenic pathogenesis. So we have to know about lignans. They are A and B, two structure which is shown here. A is phenyl propanoid unit, as I told, phenyl benzene with 4 CH2, 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 3 CH, that means 9 carbon atom, phenyl propanoid unit. Lignan structure 2 phenyl propanoid unit, that is diphenyl propanoid units. When it is joined together, very simple, 18 carbon atom joined together, you can say lignans. A student can easily remember the structure. So lignans, when food containing lignan precursors, you can say, precursor, not precursor, precursors, the in-between compound, are consumed, they are converted into entrolignans, remember, entrolignans, entrodiols and entrolactones. So what the lignans are having 9 carbon atom together, 18 carbon atom compound of an phenyl propanoid compound, lignan, which can be converted when you consume into entrolignans, entrodiols, entrolactone. Entrodiols means 2 OH groups, entrolactone means a lactone ring with O. So by bacteria, they normally colonize the human intestine. They normally colonize the human intestine. So interesting for the pathological conditions and its therapeutic application. Entrodiol and entrolactone have weak enterogenic activity in GI, but maybe exert biological efforts or effects through non-estrogenic mechanism. So non-estrogenic mechanism, they are exerting a biological effect through non-estrogenic mechanism, not directly like an steroidal estrogens of our hormone, non-estrogenic mechanism, because entrodiols, entrolactone can mimic means same, some of the effect of estrogens. Estrogens, progesterones, you may be knowing about what is its major important in the various activities. Apart from even sexual activities, apart from the sexual hormone, it has many other activities in our body, very many other pathological activity, estrogen and progesterone. So you should know entrodiol and entrolactone, a lignans which is present in the plant, have a non-estrogenic mechanism to give and mimic the action of estrogen. Their plant-derived lignans, precursor, are classified as phytoestrogens. So a question can be asked, phytoestrogens. What is phytoestrogens? You have to write about lignans, you have to write about the entrolignans, entrodiol, entrolactones. They are classified as phytoestrogens. Lignans, precursors that have been identified in human diet, including pinoracinool, lariciresinol, Secoisolariceresinol, matiresinol. So this students have to remember the lignan precursors are identified in human diet, including like pinoresinol, lariceresinol, secoisolariceresinol, mati or matiresinol and others. So you see the structure very clearly. You can have a chemical structure can be remembered, chemical structure of enterodiol. See the structure, phenyl propanoid, 9 carbonatum with 4 OH group, then they are called as enterodiol. And when this diol in the middle made into a lactone ring with O and double bond O, then you can say enterolactone. Very simple for the student. This is lignans. Lignans can be found in more than 70 families of vascular plants and have been isolated from different plant parts, exudates and resins. Biological activity of lignans are antiviral, anti-cancer, anti-prevention, that means cancer prevention, or anti-cancer prevention, anti-inflammatory, antimicrobial, antioxidant, immunosuppressive, hepatoprotective, osteoporosis prevention. So it has a wide biological activity. So a student should know about lignans. Example of podophytotoxin, a natural occurring aryl pterylene lignan. It is an aryl pterylene lignan. Is one of the most important compound due to its high toxicity and current use as a local antiviral agent. Podophytotoxin obtained from the plant of the genus Podophyllum family, Berberidaceae family, and it is a moiety, or it is a mitotox, or you can say mitotic toxin, whose derivative has been used as chemotherapeutic agent for anti-cancer. So interesting to know about podophytotoxin and to say that it is an example of lignans of aryl tetraline lignan. Food rich in lignans, the richest dietary sources is flaxseed, linseed, commonly available in the market. They contain secoisolariceresinol. That is the secoisolariceresinol is also a derivative. It is an example for lignans, but also larciresosinol, pinoresinol, not resosinol, resinol, pinoresinol, and matiresinol in good quantity. So you should know about the ligands, especially. Let's not ligands, lignans. You should remember about the secoisolariceresinol, lariceresinol, pinoresinol, and matiresinol. Lignan structure, basic lignan structure means you should know about the two structures which we have seen here. That is about enterodiol and enterolactone. Very simple, which a student can remember. Podophyllum peltatum with common names May, apple, ground lemon, family Berberidaceae, which we are using commonly. You know about this plant which we are using as a vegetable. An Indian podophyllum, it has like underground stolons and the plant and fruits which we can see in this form. Flax, Lignum usitatism, seeds obtained from Lignum usitatism and also known as flax seeds, linseed, family Linnaceae, is a fruit, and the seeds are like this, very common and very interesting after removing the seed coat. And Sesame indigum is a flowering plant in genus of Sesamum family Pedalillaceae. Very interesting, black and white are available. The sesame seeds, sesame oil, which we are commonly using. That's some example for lignans. Now in the 5-mark question they have asked carotenoids. Carotenoids, also called as tetraterpenoids, are yellow, orange and red, are organic pigments that are produced by plants and algae, as well as several bacteria and fungi. Carotenoids give the characteristic color to pumpkin, carrot, corn, tomatoes, canaries, flamingos and daffodils. You know, canaries is a bird, flamingo is also tall-legged birds, daffodils are flowers, tomato is, you know that, corn, you know that, carrot, you know that, pumpkin, you know that. They give the color for that particular fruit. Carotenoids can be produced from fats and other basic organic metabolomic buildings blocks by all these organisms. The only animal known to produce the carotenoids are aphids, spider mites, the insects, which acquire an ability to genus from fungi, and it is produced by endosymbiotic bacteria in white flies. I will show the picture of white flies also. Carotenoids from the diet are stored in the fatty tissues in animal and exclusively carnivorous animals, which eats both plants and animal, veg and non-veg, obtain the compounds from animal fat. In the human diet, absorption of carotenoids is improved when consumed with fat in a meal. Cooking carotenoids containing vegetables in oil increases carotenoids bioavailability. This is the bird which I told, the canary. The color is by means of carotenoids. Small songbird in the French family, originated from Micronesian islands. It's a beautiful singing, and this is, you know, that flamingo, a long red color, beautiful bird which is having a long leg, flamingo, or flamingos are type of very bird family Phoenicoptidae. And this is daffodil flower is common name of plant genus Narcissus, and any of individual species there are, these plants are available. Aphids, insects, they also have the production of carotenoids, a small sap sucking insects, members such as even family Aphidoideae, like this insects, like this in the, you can see the spiders and this type of insects which have the, even including the bugs. which we can see in the plants also have the production of carotenoids. White flies, we know that this is in plants, some fruits and some leaves. Below you might have seen this white flies. Carotene C40H56 is one of the most important group in carotenoids. Natural pigments which owe their color to large number of unsaturated carbon atom arranged successively. They are also known as polinic pigments. Carotene is mainly of three forms: alpha, beta, gamma. It gives carrots their color and it's associated with chlorophyll in green leaves. The most important beta carotene and forms the 4-isoprene group. They are terpenes, isoprenoid, dimethyl allyl pyrophosphate or pyropendyl pyrophosphate, which forms the structure you might have studied in your biosynthetic pathway of acetyl pathway, where the isoprene units have the main importance to form and terpenes, cholesterol, and the products of furcinyl, products of geranyl, various monoterpene, diterpene. So beta carotene is formed by 4 isoprene units and 2 cycle each of those are beta ionone. Beta carotene is a structure. We can see how the conjugated double bond system continues with an ring structure 1 and another ring structure. You can see the beta carotene and this beta ionone, where we told about the own quinone derivative of an propanone in a 6-membered structure with a conjugated double bond system. And the carotene is converted into the organism as vitamin A, by which known as the provitamin A. Other carotenes are lycopene, which is present in tomato, C40H56, a long chain, open chain, composed of 8 isoprene unit with 13 double bonds found in tomatoes, and the quercetin, a diacid, is a linear chain of 16 carbon atoms with 7 double bonds, and 4 groups of methyl form diester gentiobiose disaccharide alpha D glucose 6 beta D glucoside is in the crocin pigment of saffron. By oxidation of carotene, other pigments are formed. They are the phylloxanthins. The most important is that xanthophyllin C40H56O2, yellow color, and can be found along with the chlorophyll and the carotene in green leaves. Carotene has great importance for human health because it has medicinal properties such as reducing the chances of heart attacks, function of soluble antioxidant, and increases the efficiency of immune system, and it is used as ultraviolet protector, and taken orally has been proved that can produce the likelihood of incidence of some cancers. So interesting. Number 4, explain tracer technique. As I told, tracer technique is very important chapter in the unit 1, and they used to ask even for 10 mark question. So I explained as which we have discussed in the class. So listen, tracer techniques in that chapter, investigation of biosynthetic studies, utilization of radioactive isotopes, And in tracer techniques, use of isolated organs, grafting methods, and use of mutant strains, they have asked. So it may be asked in essay question, that's why I explained as such, which I gave in the classroom, but you can just reduce it for 5-mark question. But still, I am giving today for your lecture as a 10-mark question answer. They have asked only for 5-mark. Tracer techniques, living plants consideration as biosynthetic laboratory of primary as well as secondary metabolite. Different biosynthetic pathways which you go study. As I told, the cyclic acid pathway, acetate pathway, but there are many acetate pathway can be asked in many ways, like you can see cyclic acid pathway, meveridic acid pathway, acetate pathway, HMG-CoA pathway, isoprenoid or isoprene pathway, terpenoid pathway. All of them have the similar pathways with different precursors, different intermediate product like acetate, mevalonate, isoprene, isoprenoid, which all have been mentioned. So the student should be knowing about what is the explanation given while the explaining about the biosynthetic pathways. Various intermediate and steps are involved in biosynthetic pathways in plants can be investigated by means of following techniques. Tracer techniques, you have to write in this way. 1. Precursor product sequence, double multiple labeling method, competitive feeding method, isotope incorporating method, sequential analysis method, and also use of isolated organ can be used, grafting method can be used, and also we can use mutant strains of bacterial strains can be used for study in tissue culture. So definition, it can be defined as a technique which utilizes a labeled compound. What is labeled compound? To find out or to trace the different intermediate and various steps in biosynthetic pathways in plants at a given rate and time. Kinetic, in the rate and the time, how the change have been taken can be identified by means of labeled compound. Labeled compound means we have got the explanation using a radioisotopes labeled with the structure. Examples have been shown in the lecture. In this technique, different isotopes, mainly the radioactive isotopes, which are incorporated, that is labeled, incorporated into presumed precursor of the plant metabolites and are used as a marker and as biogenetic experiments. You can see the structure, how the mass spectrometry, the results which we have seen, the line spectra, the nitrogen, which have been made into an isotopes and radioactive isotope, and which can be easily made in the structure as I have projected by round, showing that nitrogen present in the chemical structure, can be seen in the mass spectrometry. What is its line spectra giving the M by Z, that is about 586 line spectra. For the 15, nitrogen 15, the radioactive isotope, which having 7 protons and 8 neutrons. Neutrons and protons, when there is a change in the atom, they will become the MZ, or the charge or the radioactivity of the atom will be changed. So that will be studied by means of the change in the structure and the structural elucidation in biometabolomic study. Biosynthetic study can give you a detail. The labeled compound can be prepared by using 2 types of isotopes. Radioactive isotopes, stable isotopes. Radioactive isotopes H1, carbon 14, sodium 24, potassium 42, sulfur 35, phosphorus 35, iodine 131, delay with emission of radiation. Isotopes of carbon, you can see carbon 6 proton, 6 nitrogen, normal carbon atom. 7 proton and 6 neutrons is an isotopes. 8 protons and 6 neutrons, carbon 14 is an isotope. And you can see the sodium with 11 protons and 12 neutrons, 23 sodium. 11 protons and 11 neutrons, 22 is the sodium. And 24 sodium is also their isotopes. Hydrogen, when it is one, only one mass number, that is one, only one proton. When there is one proton and one neutron, H2 isotope. Deuterium. Tritium, when there is one proton and two neutrons, like H3 mass number, isotopes are there. So now the student may be knowing what is the radioisotopes, what is the isotopes. For biological investigation, carbon and hydrogen. The metabolic studies sulfur, phosphate and alkali, even sodium, potassium, calcium and alkaline earth metals are used. For studies of protein, you may be knowing protein made up of amino acid, nitrogen. Alkaloids, amino acids labeled nitrogen can be more specific information. H3 hydrogen compound is commercially available. And we know that stable isotopes like H2, carbon 13, N15, O18. Nitrogen 14, you know that 7 protons, 7 neutrons, 7 electrons. Nitrogen 15, 7 protons, 8 neutrons and 7 electrons. And nitrogen 16, 7 protons, 9 protons and 9 neutrons and 7 electrons. So use of labeled compounds as possible intermediates in biosynthetic pathways. Usual method for detection are mass spectrometry, nitrogen 15, oxygen 18 can be done, NMR spectroscopy or spectrometry you can use, H2 and carbon 13. Significance of tracer techniques. What is its application? What is its significance? Tracer techniques of biosynthetic pathway. Example, by incorporating the radioactive isotopes of carbon 14 into phenylalanine, the biosynthetic cyanogenic glycoside prunosine can be detected. Location, quantity of compound containing tracer carbon 14 labeled glucose is used for determination of glucose in biological system. Students have to see that very clearly, my explanation regarding what is labeled compound, what do you mean by carbon atom when it becomes 14 is labeled, and nitrogen how it is labeled. I have given an examples in the next slide also. Different traces for different studies. For studies on nitrogen and amino acids, labeled nitrogen give specific information than carbon. Convenient for suitable techniques. So, criteria for tracer techniques important. The starting concentration of the tracer must be sufficient withstand the resistant with dilution in course of metabolism, natural metabolism. It should have a capacity to stand in the natural metabolism. Proper labeling. For proper labeling, physical, chemical nature compound must be known. You see a labeled compound of D-glucose with carbon 13. 6 carbon atoms have been labeled. You know that glucose molecule is C6H12O6. You see the molecule, all the carbon atom is made into carbon 13. You know that carbon 12 is normal and carbon 13 is isotope. And radioactive isotopes of carbon 13, carbon 13, carbon 13, carbon 13, carbon 13. 6 carbon atoms present in the D-glucose have been made into carbon 13. A labeled compound should be involved in the synthesis reaction. That is very important. And you can see here another example of glutamine, an amino acid, labeled with 15 nitrogen. See that 2 nitrogens are made into 15 nitrogen as I highlighted in glutamine. So this is how labeled compound. Nitrogen normally 14, it is made into 15 by means of radioactive isotopes. Labeled should not be damaged by the system which is used in the experiment. And another example of a precursor like phenylalanine, aromatic amino acid with nitrogen 15. You know the formula C6H5, CH2, CH, NH2. This N is made into 15. Isotope COOH. You see that nitrogen is made into, and pyruvic acid, the carbon atom can be made into labeled by CH3COOH. That CH3 is made, C is made into carbon 13. And another example of lithium salts of acetyl-CoA enzyme in that carbon atom. You know that C2C12 H36 is a formula of a coenzyme complex of a lithium salt. The carbon atoms are made into radioactive isotope with C13. So this is an advantage, highly sensitivity, applicable for all living organisms. Wide range of isotopes are available. More reliable, easy administration and isolation procedure. Give accurate results in proper metabolic time, kinetic and technique applied. Limitations are also there. Kinetic effects, chemical effects, radiation effects, radiochemical purity, high concentration distortion the result. So this all some of the disadvantages. How to make labeled compound? They are asking 5-mark questions sometimes. By means of growing chlorella. What is growing chlorella? We should know. Catalytical exchange, chemical reaction, organic synthesis. Labeled compound produce growing chlorella, growing in plant or growing in tissue culture in an atmosphere of carbon 14 or carbon dioxide 14. All carbon atom can be labeled by radioactive carbon atom 14. And H3 labeled compound are commercially available. Tritium labeled is affected by catalytic exchange in aqueous media by hydrogenation of unsaturated compound with tritium gas. Tritium is a pure beta emitter of low intensity. Its radiation energy is lower than carbon 14. By the use of organic synthesis, which we know about Grignard reagent CH3 MgBr with carbon dioxide 14 chlorella of making the atmosphere reacting with the carbon dioxide 14 CH3 14 COOH MgBr catalytical reagent synthesis with carbon atom with 14 with water CH3 CH acetic acid MgBr OH can be removed, and this type of synthesis also can be used for labeling the carbon atom of a precursor. Precautions. The precaution should react a necessary site of synthesis in the plant. Plant at an experiment time should synthesize the compound under investigation. The dose given is for short period. Introduction of the labeled compound. We can do how to do the experiment. Root feeding, stem feeding, injecting, direct injection and infiltration, floating method, spraying method can be followed. So these are some of the method in the and separation and detection of the compound after injection. How you will detect? Using instruments like Geiger-Müller counter, liquid scintillation counter, gas ionizing chamber, Bernstein or Bernstein counter, mass spectrometry, NMR spectroscopy, radio or autoradiography, radio paper chromatography. These all instruments can be used. Just I have added what is a Geiger-Müller counter, they may ask. It's a chamber which having an anode and electrode. Often electrodes will be present. Ionizing gas, atoms, the helium or organ which is inserted to ionize the compound, and so that you can, the Geiger-Müller counter is a device used to detect the measurement of type of radiation, alpha, beta, gamma radiation. Basically it consists of pairs of electrodes surrounded by a gas. The electrodes have high voltage across them. The gas used is usually helium and argon. When radiation enters into the tube, it ionizes the gas of argon or helium which is used there. The ions are attracted to the electrodes and the electric current is produced, and it can be analyzed by means of computerized, and the scale count the current pulses and even the count wherever radiation ionizes the gas. Scintillation counter little advanced, where you can see X and Y rays fall on an sodium crystal present in a plate, and also it can be generated by a photo cathode and multiplied by anode, and the voltage can be analyzed by computer, which is used for scanners. Scintillators are used in conventional film, screen radiography and digital radiographic receptors, fluoroscope, scintillation cameras, most CT scanners and PET scanners, positron emission tomography PET. The scintillation detectors consist of scintillators and or device such as PMT, that is photomultiplier tube with many anodes which multiplies the small rays which is coming from the ions that are converted into light in the electric signal. More sensitivity than Geiger-Müller counter. Widespread detection can be done by scintillation counter. Principle of scintillation counter is incident radiation intact with the material. Atoms will rise to excited state. Excited states exhibit and visible light fluorescence, and the light strikes on the photosensitivity plate of which is kept as a cathode and anode, and photosensitivity generates the electricity, releases the photoelectrons multiplier. Autoradiography, detection of radioactive compounds with photographic emission, X-ray film. Types: in vivo autoradiography. Receptors are labeled in intact living tissues by systemic administration by the radioligand, PET. Positron emission tomography. In vitro radiography, that is in vivo and in vitro radiography. Slice mounted tissue, sections are incubated with radioligand, so the receptor are labeled under very controlled conditions. So it can be done in vivo and in vitro. Uses: map anatomical location of radio labeled ligands to visualize the quantified receptor in tissues. Trace neurons and oxanols transport of radioactivity labeled amino acids, certain sugars are transmitter substances. Measure DNA production, example of H3 thymidine. So in in vivo radiography, we are having scanners. When the radioactive isotopes in a ligand decay, it can be measured by positron emission tomography on single photo emission, which we are using for CT scan. And in animal, in vitro, directly in the living cell, chiral selection of the tissue, radioligand equilibrium binding, and then exposure to the imaging scanner, phosphor and digital autoradiogram, and quantification can be done. Mass spectrometry is an analytical technique that measures the mass to charge ratio, that is M by Z, relative abundance of the ion charge particles. It is used to determine masses of the particles for determining the elemental composition of the sample or molecules, and for elucidating the chemical structure of molecules such as peptides and other chemical compounds. NMR spectroscopy is a research technique that exploits the magnetic property of the certain atomic nuclei to determine the physical and chemical properties of atoms or the molecules in which they are contained, which we have detailedly explained about NMR spectrometer separately. It relies on the phenomenon of the nuclear magnetic resonance, can provide the detailed information about the structure, dynamics, reaction state, and the chemical environment of the molecules. Methods of tracer techniques, just now we have seen about what all the methods. Number 1, precursor product sequence. Number 2, double multiple labeling. Number 3, competitive feeding. Number 4, isotope incorporation. Number 5, sequential analysis. And we can go for use of isolate organs, and you can go for grafting methods and use of even mutant strains. This is important for the 5-mark question what they have asked in December 2023. Whichever I have given is for an essay question, you have to add the previous section. This is very important for you to write this 5-mark question. Precursor product sequence means it is a technique of presumed precursor of the constituents under investigation on a labeled form is fed into the plant, and after a suitable time the constituent or the active principle or the precursor is isolated, purified, and radioactivity is determined. Disadvantages: the radioactivity is isolated compound alone is not usually sufficient evidence that particularly compound fed in directly precursor, because substance may enter into the general metabolic pathway and form other products. There may become randomly distributed throughout the whole range of the product. Application of precursor product sequence: stopping of herdinine production in barley, seeding after 15 to 20 days of germination. They have done this work. Restricted synthesis of hyoscine and from hyoscyamin in Datura stramonium. This method is applied for biosynthesis of morphine and ergot alkaloids. Double and multiple labeling. This method given evidence for nature of the biochemical incorporation of precursor rise double and triple labeling. In this method, specifically labeled precursor and their subsequent degradation to recover product are more employed. Application of double and multiple labeling. This method is extensively applied to study the biosynthesis of plant and secondary metabolite used for study of morphine alkaloids. Double and multiple labeling it is done by Dr. Lenti use a double labeling lysine, example, with carbon 14 and a specific position fed in Nicotiana gluca plant to trace an origin of pyridine ring. Nicotine is made up of pyridine ring in the alkaloid anabasine, how they demonstrating with the hydrogen atom from lysine and incorporating proper lysine role forming a ring structure to give the piperidine ring structure. So the basic application of the isotopic tracer technique is natural product biosynthetic study. You can see this reaction. The labeled carbon atom near to the NH2 group in a lysine in the plant, and the nitrogen is also labeled, carbon is also labeled, and they found that what is the product they obtained, and we are getting both the compound is labeled in anabasine, and they sent with the carbon atom labeled and they got the carbon atom labeled. So this is how double multiple labeling, two compounds carbon and nitrogen can be labeled separately, nitrogen carbon alone can be labeled, and find out what is the product is obtained from the plant by injecting, and we can see the labeled compound change. Competitive feeding, which one, whether B or B dash is the precursor in between responsible, as shown in the picture, A to C, whether it comes through B or whether it comes to B dash. So they are finding out by means of giving a labeled compound in the A and finding out what type of compound, whether it is in between compound B or B dash. If the incorporation is obtained, necessary to consider whether this is fact the normal route of the synthesis of the plant, not this subsidiary pathway. Competitive feeding is distinguished whether B or B dash is the normal intermediate in the formation of C from A. Application, competitive feeding, this method is used to elucidate the biosynthesis of propane alkaloids and biosynthesis of hemlock alkaloids like choline, conhydrin, etc. Biosynthesis of alkaloid of conium maculatum, that is hemlock, using C14 labeled compound. You see the structure here, which we can see the lysine, which is coming through choline or gamma cholicin to give an conhydrin. It can be easily found out what is the main intermediate competitive feeding, which one, that is whether conilin or gamma cholicin can be identified to give a lysine giving the labeled compound, labeled carbon atom, and which we can find out polybeta keto acid, which undergo gamma cholicin to give a conhydrin in hemlock. That is, hemlock alkaloids can be studied, an example for competitive feeding. So isotope incorporation method. In this method, provide information about the position of the bond cleavage, their formation during reaction. Example, glucose 1-phosphate cleavage as catalyzed by alkaline phosphate. This reaction occurs with cleavage of either carbon-oxygen bond or phosphorus-oxygen bond. So you can see here the glucose molecule phosphatase and how they are, and how you are getting the water molecule with oxygen 18, and where it has been changed by means of phosphoric acid can be studied by isotope incorporation method. And sequential analysis, the principle of the method is investigation is grow a plant in atmosphere of carbon 14, then analyze the plant at given time interval in kinetics and obtain a sequence in which various correlated compounds become labeled. Application of carbon dioxide 14 sequential analysis has been very successfully used in elucidation of carbon and photosynthesis. Determination of sequential formation of opium, hemlock and tobacco alkaloids. Exposure as less than 5 minutes. Carbon dioxide 14 is used as a detecting biosynthetic sequence as pipritone, methone, menthol and mentha preparator. An application of tracer technique, Study of squalene cyclization by use of carbon-14 H3 labeled mevalonic acid. Interrelationship of among 4-methyl sterol and 4,4-dimethyl sterols by use of carbon-14 acetate. Terpenoid biosynthesis by chloroplast, isolated in organic solvent by use of 2-carbon-14 mevalonate compound, and it can be identified. Study the information or formation of cinnamic acid in pathway of coumarin from labeled coumarin. So this is the two questions. An application continues with the origin of carbon, nitrogen atom, purine ring system, carbon 14, nitrogen 14 labeled precursor. Study formation of scopa, that is scopolamine used in labeled phenylalanine. Aromatic amino acid can be used. By use of calcium 45 as a tracer, found uptake of calcium in plants from calcium oxide and calcium carbonate. By adding ammonium phosphate labeled with phosphorus 32, known specific, it's a specific activity and uptake of phosphorus is following by measuring the radioactivity as labeled reaches. First in low parts of the plant and then upper part of the plant, leaves, etc. can be done. So other methods used in biosynthetic investigation, we know that isolated organ, tissues and cells. We can isolate the tissues and the tissue culture, plants and as well as animal tissues, grafting, and we can cut one plant and fix with another. Like grafting techniques are now very common. In homely it can be possible. Instruments, plastic and synthetic products are also available. Mutant stains, these three. Other than tracer techniques in the biosynthetic investigation can be done. So by mutant strains also, bacterial growth, fungal growth, if the tissues can also be made. Isolated organ, the method is based on using isolated parts like stem or root. The technique is useful to determine the site of biosynthesis in particular compounds. Roots and the leaf are studied Nicotiana, Datura, petal disc for study of rose oil, tropane alkaloids in root of Solanaceae family. Grafting, the method is used to study alkaloid formation in grafting plants. Tomato sessions grafted in Datura producing alkaloids, while Datura session grafted in tomato produces less quantity of alkaloids. A simple example. This shows the main site of biosynthesis in the root. So this is one picture of how they are doing the grafting by means of with the stem part, with the root part or the stem part of the other plant. And use of mutant strains, it is a mutant strains of microorganism produces the lack of certain enzymes. Example of Gibberella mutant used to produce isoprenoid compounds. Lactobacillus acidophilus is used for mevalonic acid pathway for isoprenoid biosynthesis. Enzymatic study, phytohormone, auxin plays critical role in a relationship of plant growth and development. Indole 3-acetic acid IAA has been recognized as a major auxin which is synthesized in the plant is still unclear. Previous genetic and enzymatic studies demonstrated that both tryptophan and aminotransferase of Arabidopsis TAA and the YUC family, that means in flavin monooxidase like proteins are called as YUC, and requires to biosynthesis of IAA, indole acetic acid, during plant development. Auxins, gibberellins, hormones, responsible for the development of the plant can be studied by means of mutant strains. The TAA, that is, for example, tryptophan amino acid family produces indole 3-pyruvic acid, IPA, and YUC, the family flavin, monooxidase like protein function, is conversion of IPA, indole pyruvic acid to indole acetic acid in Arabidopsis kaleena. So these are some of the uses of mutant strains which can be used. Arabidopsis kaleena, this is the flower, is used for this mutant strain study. And this study can be given even as cellular changes and micro ATPs changes, which can be also be done by means of various, that is, laboratory method, which can be done in vitro study. And we will continue with the short answers, and the brief answers are there in the next lecture. Thank you for being for this lecture. Listen, try to make a hints and write down your own notes. That will be more helpful for you. Thank you for listening. We will continue this short questions again, like describe the anatomy of digitalis and all in the next lecture. Thank you for listening this lecture.